You know that feeling, right? When your favorite pants suddenly feel just a little bit snugger around the waist.
Oh, yeah. Happens to a lot of us.
It's kind of a classic sign of getting older, isn't it?
Yeah.
But it turns out that expanding waistline is, well, it's more than just a cosmetic thing.
That's a really crucial point. It seems like scientists are increasingly finding its link to, the whole aging process maybe speeding up a bit and our metabolism definitely slowing down.
Right. What we might just see as, annoying weight gain is actually tied to these deeper biological processes.
Exactly. And that fat accumulation, particularly around the abdomen, it's a big factor in age-related health risks. No doubt about it.
So what's really going on? I think most of us just assumed, well, our existing fat cells just get bigger over time.
Yeah.
Simple as that.
That was the prevailing thought, yeah.
But today we're doing a deep dive into some really fascinating new research. It's from scientists at City of Hope, and it was published in Science. It suggests there might be a whole different mechanism at work.
A really different mechanism.
So our mission here is to unpack that, to understand the biological reasons behind this age-related belly fat based on this new science.
And it's quite a shift in thinking. This new work suggests it's not just about existing fat cells expanding. They've actually identified, believe it or not, a brand new type of stem cell.
A new stem cell.
Yeah, one that seems to be a key driver in this whole process.
Okay, so we thought the issue was just our old fat cells puffing up. But these researchers, they suspected something else. Maybe our bodies are actually creating new fat cells as we age, specifically there in the belly area.
Precisely. They started looking closely at what's called white adipose tissue, or WAT.
WAT, right. That's the
Yeah.
main fatty tissue we associate with weight gain.
Correct. And within this WAT, there are stem cells. They're called adipocyte progenitor cells, or APCs for short.
APCs. Got it.
And these APCs, they have the potential to turn into mature fat cells. That's their job, basically.
So the researchers wanted to see how these APCs behave differently in older versus younger individuals. And they used mice for this, right? Some pretty clever experiment.
Incredibly clever, yeah. What they did was fascinating. They took these APCs from older mice and transplanted them into young mice.
Okay.
And then they did the reverse, took APCs from young mice and put them into older mice. A direct swap.
Ah, okay. I see where this is going. What happened?
Well, the results were pretty stark. The APCs taken from the older mice, when put into the young mice, they just took off. Started rapidly producing a whole bunch of new fat cells.
Wow. But what about the young cells in the older mice?
That's the key part. When they put the young APCs into the older mice, they didn't see that same surge. Not much new fat cell production at all.
So it wasn't the age of the mouse host, the environment.
Exactly. It showed that those older APCs had some kind of built-in intrinsic ability to churn out new fat cells. Something fundamental had changed within the cells themselves as they aged.
Okay, that's a huge finding. So how did they figure out what changed inside those older cells?
That's where the really high-tech stuff comes in. They used a technique called single-cell RNA sequencing.
Ah, right. That lets you look at the gene activity inside individual cells, doesn't it? Not just an average of a whole tissue sample.
Precisely. Gives you a really detailed picture, cell by cell, so they could compare the gene activity profiles of APCs from young mice versus those from older mice.
And what did that incredibly detailed look reveal? Was it what they expected?
Well, this is where it gets kind of counterintuitive, actually. We often think of cellular processes slowing down with age, right?
Yeah, winding down.
But here, they found that the APCs in the middle-aged mice showed a significant increase in gene activity compared to the young ones.
An increase? In what kind of activity?
Specifically, the gene activity linked to the processes involved in making new fat cells. It was like these particular precursor cells were actually ramping up their fat production potential with age.
That is surprising. And didn't one of the lead researchers, Adolfo Garcia Ocana, have a quote about this? Something about aging unlocking potential.
Yes, he summed it up really well. He said something like, while most adult stem cells' capacity to grow wanes with age, the opposite holds true with APCs. Aging unlocks these cells' power to evolve and spread.
Wow, unlocks their power. That's quite a phrase.
It is. And he continued, this is the first evidence that our bellies extend with age due to the APC's high output of new fat cells. It really highlights how unexpected this finding was.
So this heightened activity, this discovery, it led them to pinpoint a whole new type of stem cell that seems to pop up as we get older.
That's right. They gave it a specific name, committed preadipocytes, age-specific, or CPAs for short.
CPAs. Committed preadipocytes, age-specific.
Okay.
And committed means they're already destined to become fat cells.
Exactly. Unlike the more general APCs, which might have other potential fates, these CPAs are pretty much locked onto the path of becoming fat cells, and they seem to appear mainly in middle age.
And these CPAs are the real drivers of the new fat cells in older mice.
That's what the evidence points to. They are highly efficient at generating new fat cells, explaining that increased fat accumulation they saw.
Okay, so we have these CPA cells actively making new fat, but what tells them to do it? Is there a trigger, a signal? The research mentioned a specific pathway, I think.
Yes, they identified a crucial signaling pathway. It's called the leukemia inhibitory factor receptor pathway, or LIFR.
LIFR, okay.
And it appears that this LIFR signaling is absolutely essential for these CPA cells. It helps them multiply, and it helps them mature into actual fat cells.
So without this LIFR signal,
The CPAs wouldn't be nearly as effective at creating that age-related belly fat, it seems.
and Annabelle Wang, another researcher on the study, she explained this quite clearly too, didn't she?
She did. She said, we discovered that the body's fat-making process is driven by LIFR. And interestingly, she noted, while young mice don't require this signal to make fat, older mice do.
Ah, so it's like a switch that gets flipped on or becomes necessary with age.
Seems like it. She concluded, our research indicates that LIFR plays a crucial role in triggering CPAs to create new fat cells and expand belly fat in older mice. It highlights a new dependency that develops with aging.
This is all fascinating stuff in mice. But, the big question is always, does this apply to us? To humans? Do we have these CPA cells too?
The million-dollar question, right. And encouragingly, yes. The researchers did look at human tissue samples.
Oh, good. What did they find?
Using that same single-cell RNA sequencing technique on human fat tissue taken from people of different ages, they found cells that looked remarkably similar to the mouse CPAs.
Really? Similar characteristics?
Yes. And importantly, these human CPA-like cells also showed a high capacity for generating new fat cells. And just like in the mice, they found more of these cells in samples from middle-aged individuals compared to younger ones.
Okay, so it looks like this mechanism isn't just a mouse thing. It's likely relevant to human biology, too.
It strongly suggests that, yes.
So, if these CPA cells, driven by this LIFR pathway, are a key reason why many of us gain belly fat as we age, what are the potential implications, thinking down the road, for health?
Well, the implications could be pretty significant if we can figure out how to control the formation of these specific new fat cells.
Maybe by targeting the CPAs directly?
Mm-hmm.
Or that LIFR pathway?
Exactly. If we could do that, it might give us a completely new way to tackle age-related obesity. And since we know excess belly fat is linked to so many health issues, diabetes, heart disease, you name it.
This could potentially lead to ways to improve overall health span, maybe even longevity.
That's the hope. It's about addressing a fundamental aspect of metabolic aging.
It definitely sounds like this opens up some really exciting avenues for future research. What are the scientists planning next? Where does this go from here?
Right, so the immediate next steps involve tracking these CPA cells more closely in animal models, really understanding their life cycle, how they behave over time.
Getting a clearer picture.
Yes. And of course, continuing to study these cells in humans. More samples, different populations, really confirming their role.
Makes sense.
And then the longer-term goal, obviously, is to explore therapeutic strategies. Can we find ways to safely and effectively eliminate these CPA cells? Or maybe block that crucial LIFR signal just where it's causing problems.
Lots of work ahead, but very promising.
Definitely.
Okay, so let's try and boil this down. For everyone listening, what's the single most important takeaway from this deep dive today?
I think the key thing is this discovery. These specific cells, the CPAs, that emerge with age, and the realization that the LIFR pathway is critical for them to make new fat. It gives us a concrete biological reason, a mechanism for why that belly fat tends to creep up as we get older.
It's not just vague slowing metabolism anymore. It's something specific we can potentially target.
Exactly. It shifts the understanding.
And importantly, this isn't just about vanity, about fitting into clothes. This research could genuinely pave the way for future medical interventions, couldn't it?
Absolutely. By understanding these basic cellular mechanisms of aging fat tissue, we really open the door to thinking about new therapies. Therapies aimed at preventing or maybe even reducing age-related obesity and all the health risks that come with it. It's fundamentally about promoting healthier aging.
So here's something to chew on as we wrap up. When you think about this research, consider the bigger picture. What does it really mean to understand these tiny, specific cellular changes that drive aspects of aging? How might knowing this stuff, knowing why our bodies change in these ways, influence not just how we approach weight gain, but maybe our entire strategy for staying healthy and preventing disease as the years go by? It could reach far beyond just thinking about our waistlines, couldn't it?